Hi, good evening. My name is Khadija and I am your tutor for this program. We are going to be starting with the embryology of the nervous system, and in your course module you're supposed to be starting with the spatial senses, that is the tongue, the eye, the nasal cavity, and also the auditory system. And then you're supposed to also talk about the endocrine system as well as the central nervous system. But I feel like it's better for us to start with the model 3 so that you understand the development of the nervous system and the nervous system itself, such the way that when we start talking about the special senses, we would not be strangers to the different parts of the brain where the neurons of those special senses are going to travel, and then their own central areas where they need to take this information and interpret this information. So in the model 3, which is central nervous system, we're going to talk about the embryology, which is the development of the nervous system, and then we'll also talk about the cerebral hemispheres and brainstem, that is study 2. Study 3 will be talking about the diencephalon, basal ganglia, the ventricles, and cerebrospinal fluid, while study 4 will talk about the spinal cord and the blood vessels of the nervous system completely. So the central nervous system, the nervous system is comprising of the central nervous system and peripheral nervous system. So the peripheral nervous system is a system where is a system that contains the nerve fibers. So in this illustration, and the yellow color is an indication of the peripheral nervous system. So these are nerve fibers that carry information from either the brain, or the spinal cord. So the brain and the spinal cord are central nervous system, while the yellow fibers leaving the brain and the spinal cord are peripheral nervous system. So they take information to the exterior or the effector organs, and also perceive information from the body wall and take it back to the brain. So these information taken back to the brain are mostly sensory innervations and are called efferent, while information leaving the brain or the spinal cord going towards the exterior to execute a function, whether move our hand away from a pin, and that is called an efferent fiber. So these peripheral nervous system are also further classified as autonomic nervous system, that is the ANS, and then the somatic nervous system. So the somatic nervous system is a system that we can actually control, like moving our hand, but the autonomic nervous system is a system that we do not have a control over it. It automatically does its function. Like we're sitting down here, we cannot control our heartbeat. It's because it is being controlled by the autonomic nervous system. Our glands are producing saliva not by our control, but by the control of the autonomic nervous system. And sometimes when we cry, we are not controlling our lacrimal glands because they are also controlled by the autonomic nervous system. So this autonomic nervous system is subclassified into parasympathetic, sympathetic, and enteric. Alright, so enteric is where it controls the digestive system. It's just at a rest. Our systems are moving on their own. The heart beats and also the diaphragm controlling the expansion and relaxation of our lungs. So when we talk about what a parasympathetic nervous system is, so parasympathetic nervous system is for rest and digest. So for instance, if right now there was a fight or an accident, the fear we have at that point when we either run towards it or run away from it. So most of the time in textbook you hear the fight or flight. You either fight it or run away from it, that is flight. But it's when you go and stop running, that is when you try to process, you rest and digest this information, and that is where you have a response. You either cry or get anxious, so that is parasympathetic. So sympathetic is actually made to fight or to run. That is where we say the flight or fight, and the parasympathetic is rest and digest. So let's go back to the central nervous system, which we'll be discussing more of it in this section, where it's the central nervous system is comprised of the brain and the spinal cord. So the brain is also subdivided into the three parts. We have the forebrain, the midbrain, and also the hindbrain. So when we say the forebrain, we're talking about the cerebral hemispheres. The midbrain, we're talking about the brain stem. And also we're talking about the hindbrain, where we're talking about the cerebellum, which is a small brain which is involved with our balance. Then next we are going to be seeing the spinal cord. So the spinal cord is this elongated structure that runs from the base of our brain all through our spinal vertebra. That is our spinal cord. And this spinal cord also, we have the exits of yellowish structures leaving the brain and also the spinal cord. So the ones leaving the brain, we have, it's called the cranial nerves, and then the spinal nerves as the one leaving the spinal cord. And so here they interchanged it actually. So we have 12 cranial nerves and 31 paired spinal nerves. So the central nervous system is the commander of the body, is responsible for processing information and coordinating action. And like we saw earlier on, it's separated into the brain and then also to the spinal cord. Alright, so this is a cross section through the spinal cord, and there you can see the gray matter, and the spinal cord is located in each side in the central aspect, which is the grayish color, while the exterior is more whitish in color. The reverse is the case in the brain. Both the brain and spinal cord are also covered by what we call the dura mater. And also we have 3 layers, so they are called the meninges. We have 3 layers of the meninges found around the brain as well as the spinal cord. These 3 meninges comprises of the dura mater, which is the hard exterior, and then deeper inside to the dura mater we have a spider web looking structure which is called the arachnoid mater, and directly lying over the spinal cord and the brain is what we refer to as our pie matter. This we're going to see later on. So the brain proper is divided into several parts, and the main function is controlling our thought, emotion, movement, and sensation. And we said it's divided into, so this, when we talk about the brain, we're talking about the cerebral cortex, which is the large brain. So we have the frontal lobe, which is responsible for making decisions. So we make our decision, and also some problem solving is also in the frontal lobe, where if you choose to either walk away or walk towards something, the frontal lobe does that. Then we have a part of the brain which is most where we can you can put on top of the middle of your head, is referred to as the parietal lobe. This is involved with processing sensory information, including touch, temperature, and pain, and also proprioception. So proprioception is the movement of our joint. And then we have, on the most posterior aspect, we have the temporal or the occipital lobe, which is involved in processing the visual information. And on the lateral side of our brain, we have what is referred to the temporal lobe, which mostly involve the interpretation of hearing, language, and memory. So this is what we just explained. These are the different lobes for this bigger part of the brain. So this is the bigger brain. At the bottom of it, we have the small brain, or which we refer to as the cerebellum. But the bigger brain is called the cerebral cortex. And the cerebral cortex is subdivided into 3 different parts, like we said in the previous slide. So we have the frontal lobe, the parietal lobe, and the yellow is telling us the temporal lobe, which is found by deep to our ears. And behind our head, we find the occipital lobe, which is involved for our vision. And that's why some people, when they have problem with their sight, the back, most of the time they have headaches coming from the back of their head. And this is because the occipital lobe is involved with interpretation of our visions. And also the parietal lobe, which is this deep purple color, is involved with our sensation and how to interpret what we sense, the sense of touch, temperature, or pain. While the frontal lobe, like we said, is involved with initiation of problem solving, etc. So on the spinal cord, we said, is something that is a long bundle that extends from the base of the brain all the way to the vertebral bone, and at it stops at the actual brain stops at the level L1, L2, and it finishes as a cone looking structure, and then the other parts of it, the lower part of it, continues with the neuronal fibers of the spinal cord. This is very important clinically, which we'll discuss later. So the spinal cord runs from the base, that is where we have the foramen magnum, that is where the spinal cord lives, and then extends all the way. It's almost suspended in the vertebral column, but it's also held on by the dura mater and also some suspensory ligaments, that including the denticrete ligament. And then it all extends all the way to L2, L4 level of the lumbar vertebra, and then from the lumbar vertebra all the way to the tip of the coccyx, we have like a broom or a horsetail looking collection of fibers, which are the nerve fibers at that end. So the collection of that nerve fibers at that end is referred to as the cauda equina, and this is important in clinical aspect because it serves as a point where one can actually do a collect injection, where you can actually take a sample of the amniotic fluid for testing, or is a point where you can do an anesthetic injection such as the epidural injection. And there, if you decide to prick that area, you are not going to harm any spinal cord, because any injury to the spinal cord may have significant effect in the function of that particular area where the nerve the nerve fiber of those structures are located, and which could be associated with different types of paralysis. So the brain itself is covered by several layers of structures. From the most exterior, we have the skin and the scalp, and also the connecting tissues are what is mostly referred to as the scalp. And then you have the bone. So covering the bone is called the periosteum. So this is found both protecting the brain and the spinal cord. So the skull protects the brain, while the vertebral body protects the spinal cord, which runs in the vertebral column. And next to that, deep to that, we have what are referred to as the meninges. And like we said earlier on, the meninges have three layers. So the most exterior layer, lining just beneath just deep to the skull, is the dura mater. And dura mater is a thick layer of matter, is very tough, and we have double layer of that, which is important because the folding of that double layer will give rise to something known as the sinuses, which serve as a blood drainage system of the brain. So the brain, it doesn't have like a vein proper, but rather the venous drain the venous drainage of the brain is done by the sinuses in the brain. Then deep to the dura mater, we have the web, the spider web looking area, which is called the arachnoid mater. And the arachnoid mater has also a space deep to it, which is called the subarachnoid space. And after the subarachnoid space, we get to see the thin layer of covering that covers directly onto the brain or to the spinal cord, and this is called the pia mater. Deep also found inside the subarachnoid spaces are mostly the blood vessels, which are mostly the arteries supplying the brain, as the venous drainage is through the sinuses. Next, we have the cerebral spinous, which are also, like I said, are double folds of the dura mater, and they serve as a venous drainage to the brain. Another protective layer is called the cerebrospinal fluid, So the cerebrospinal fluids are found inside the ventricles. So ventricles are air spaces found deep to the cerebellum and also down behind the what we refer to as the brainstem. So behind the brainstem there is a space before we see the small brain, which is the cerebellum. So between these two structures there's also another space, and this space will run all the way down through the spinal cord via the central canal. So outside this spinal cord you also have the fluid. You remember we have that subarachnoid space. So the cerebrospinal fluid is being produced in the ventricles, and it also moves inside these opening spaces and goes into the subarachnoid spaces, where it gets drained into the blood vessels that are found deep into the cerebral cortexes. So another important thing that serves as a protection to the brain are the blood-brain barrier and the cerebrospinal fluid barrier. So the blood-brain barrier is a communication or a junction where it is between where the blood vessels lie onto the brain. This serves as a selective point between the brain structure and the blood, where the blood vessels carry selected nutrients from the blood and gets diffused into the brain, because we do not want any big harmful structures to cross these blood vessels to enter onto the brain. And that there is a special communication between the blood vessels and the brain, and that allows for selective passage of structure, and that is why it is called the blood-brain barrier. So we are going to discuss this in details later on. The second one is also referred to as the blood cerebrospinal fluid barrier. This is found between the communication of the blood vessels as well as the ventricular system, where we had the ventricles, which are the openings or cavities found deep inside the brain. So these also ventricles are lined specially by some cells, which also prevents the crossing of structures from the blood vessels to the ventricles. And so this is important because this is found where we have the production of the cerebrospinal fluid. So this also serves as a selective point where only selective structures comes in from the blood vessels into the ventricles via the endothelial cells of the ependymal cells of the ventricle. So now in details, like we said, the scalp and the skin are very important, like I've seen in the image on the right hand side, where you can see the skin is shown, and then we have the periosteum and the skull bone. After that we have the two layers of the dura mater, so you can see one is in gray and one is more like a pink color, because you have a periosteal layer of the dura mater, and then you have the deeper layer which is called the visceral layer of the dura mater. The visceral layer of the dura mater is the one that projects in between the folds and the brain, which we're going to talk about later on. So the folds of the brain also have their names. So we see that the visceral layer of the dura mater is the one that projects into the fold. The next layer, like we said, is the arachnoid matter, and between the arachnoid matter and the pie matter. So the pie matter is the one directly covering the tissues of the brain. So between that two space, we have the subarachnoid space, which carries the blood vessels in that particular area. This is also a similar appearance in that of the spinal cord, where the brain, after the body of the vertebral bone, deep inside the canal, we have the dura mater and the two layers of the dura mater as well. Now, most of the time here we have one layer of the dura mater in the spinal cord, and then We also have next to the dura mater, we have the arachnoid mater, and directly lining the spinal cord is the pia mater. And so sometimes at the dura, some points of the dura mater, you get to see some projections of the dura mater going onto the wall to hold onto the vertebral wall, as well as onto the pia mater to send like a suspensory support to the spinal cord that is suspended inside the ventricles. So here, like we said, deep here we have the meninges. So the meninges is the periosteal and visceral layer of the dura mater, and that is like shown in this image. So if you check, you can see the dura mater, arachnoid mater, and the pia mater and the spinal cord as well. Alright, so I like to show different images. So here is also a representation of the layers in the brain. So the one on the left is the one shown in the brain, while the one on the right is showing the cross section or superior view of the spinal cord, where you get to see the spinal cord inside the vertebral bone, and then you can see the different layers, including a fat part surrounding the spinal cord, which helps to reduce the suspension, the free suspension of the spinal cord in the vertebra. And then you can see externally, you can see the dura mater, which is more reddish in color, and then you can see the arachnoid mater, more blue in color. The pia mater is firmly attached onto the spinal cord, and that is why we don't get to see it very clearly. So like we said, that the folds of the dura mater give rise to the venous and drainages, which are referred to as the sinuses. And later on, we're going to talk about the sinuses. They are very important. They are found within the double fold of the visceral parts of the dura mater in the brain. So we have the most superior one is called the superior sagittal sinus, and then lower to it, because this is found between the right and left lobe of the brain, and that depression between the right and left lobe of the brain is called the superior sagittal fissure, and also this particular sinus is also referred to as the superior sagittal sinus. Then below here you also have another one just at the deeper part of that fissure, which is also referred to as inferior sagittal sinus. So the two sagittal sinuses are going to be linked at the trochlear, and at the points referred to as the trochlear, via a straight sinus, and then from that area they move via the transverse sinus, and from the transverse sinus some of the venous drainage from the cavernous sinus here in the middle, that mesh looking area, will communicate with the transverse sinuses via the superior and inferior petrosal sinuses, and then they all drain into the sigmoid sinus, and from the sigmoid sinus they drain into the jugular vein. From the jugular vein, internal jugular vein, they drain into the superior vena cava and into the right atrium of the heart. So before we move on, let's briefly talk about the development of the brain. Let's understand where the brain is developing from. So in this image, we are seeing how the sperm cells is fertilizing an egg, and what happened is that these particular tiny cells located outside the big egg, they are called the corona radiata. What they do is, the moment the head of a sperm cells tend to attach onto the zona pellucida, that is the white disc structure around this particular egg. So the moment it crosses that, what it does is it triggers a reaction that prevents other sperm cells from fertilizing this egg or penetrating this structure, which is referred to as the zona pellucida, this glass-looking structure. So it allows only one egg, one nucleus of a sperm to come in. So immediately one nucleus of the sperm comes in, its genetic material will fuses together with the that of the egg. So note that during meiosis, and the genetic makeup of a gamete cells has been reduced from a diploid to a haploid, and that is why at the zygote, where the two male and female nuclei fuses together, they give rise to what is a zygote. So each of the new male nuclei and female nuclei have 23 chromosomes, and when they fertilize or fuses together, they give rise to a zygote, which is now a 23 paired chromosome. So initially the gametes were in 23 chromosomes each, but when they fuse together, they give rise to a 23 paired chromosome, which is in the zygote. So gametes only have haploid, that is 23 chromosomes, and this was achieved during meiosis and when the gametes were developing. So when the gametes were developing, the genetic makeup of the cells were reduced from a 23 paired chromosomes to 23 pair chromosomes only. So these 23 chromosomes are needed for fertilization, such that we have a zygote that has 23 paired chromosomes. We don't want a zygote that has more than 23 paired chromosomes, which, if it happens, it could lead to spontaneous abortion. And that is why when one sperm cell is able to penetrate this glass structure surrounding the egg, okay, what happened is something referred to as a cortical reaction, and this cortical reaction would prevent another sperm cells from able to fertilize the egg again, and this way it has reduced the tendency of having multiple sperm fertilizing one egg. So that terminology is referred to as polyspermy. So polyspermy is avoided or prevented by the existence of the zona pellucida that is being induced by the cortical reaction. So after that, a rapid cell division occurs, and so what happened is, the moment we have the zygote, within few hours the zygotes undergo several stages of mitosis. These cells divide into 2 daughters, 4 daughters, 6 daughters, etc., and all the way until it starts to become 16 daughter cells to the 32 daughter cells. At that stage, what happens is the structure becomes as morula. So morula, it means that the external layer of the cell is now smooth, and while the inner layer, like seen here where we saw the blastocyte, this is a cross section into that is the image on the bottom, is a cross section into the egg, where we see what is happening inside the egg. So now the egg, which is now fertilized to a zygote, has a structure. The inner cells are moving to one pole. So these particular inner cells that have moved to one pole are called the inner cell mass, while the external layers of cell that have arranged themselves and they are smooth, they are referred to as a trophoblast. So at this point, what happened is that the zona pellucida surrounding it initially tends to break or hatch. It's just like egg hatch its shell. So this is what is happening. The egg will start hatching itself because it needs to enter the uterus and implant. So it means that this zona pellucida was important in the fallopian tube to avoid several things from happening. One, the zona pellucida was intact in the fallopian tube to prevent the increase in the size of the zygote, because the cells in the zygote are rapidly dividing. But we don't want fertilization to occur in the fallopian tube, and that is why the zona pellucida is there to avoid the increase in the size of the zygote. So, and another thing is that the moment the zygote is no more in the zona pellucida, it's going to implant itself to the nearest wall, and we don't want implantation to occur in the fallopian tube, because the fallopian tube does not have enough stretchy muscles that will allow for the expansion and the baby will stay. So it means if there is a fertilization, abnormal fertilization in the fallopian tube, when the embryo start to grow, it means the fallopian tube will be stretched out until it cuts off because it doesn't have enough muscular structure to hold it. So this abnormal implantation can cause the cutting off of the fallopian tube, which can lead to internal bleeding and definitely to death. So that abnormal implantation is referred to as ectopic pregnancy, and we do not want to have ectopic pregnancy, especially in the fallopian tube, because it is very fatal. So these are two of the important reasons why the zona pellucida is still intact while the embryo is still in the fallopian tube. So it takes this fertilized embryo to move from the ampulla, because fertilization occurs in the ampulla of the fallopian tube. So from the egg to move from the ampulla, fertilized zygote, to move from the ampulla to the uterus itself, it takes roughly about 6 to 7 days, and that is why at the end of the 6 or sometimes early day 6, the fertilized zygote has arrived the uterus, and then the zona pellucida will hatch off, and when it hatches off, it starts to implant. And implantation is important because that way and the endometrial wall, the cells of the endometrial wall are broken down and they release a hormone. This hormone is very important. This is important because it helps in maintaining the pregnancy. This hormone is referred to as the human chorionic gonadotrophin. Human chorionic gonadotrophin. This human chorionic gonadotrophin will go via the bloodstream and goes to the ovary of the woman. Because if you remember your biology, the moment the egg is being expelled from the ovary during ovulation, the follicular cell or sac that was holding the ovary during development will become the corpus luteum. And if the monthly cycle occurs, if there was no fertilization, what it means is at the end of our average 28-day cycle, the menstrual blood will happen. The menstrual blood happens because corpus luteum would degenerate and become corpus albicans, and when it degenerates, it means that estrogen and progesterone will not be released. And if estrogen and progesterone are not released, the endometrial wall will erode or will shade itself. It will break down and pour out, and the blood vessels that is going to break during that breaking down will lead to the bleeding. So this is what happens to our monthly cycle. However, if fertilization occurs few days, 2 to 3 days after ovulation, then it means that cycle, that eroding of the endometrial wall will be prevented by the human chorionic gonadotrophin that was produced by the implantation, during implantation. So this human chorionic gonadotropin released by the endometrial wall will go and tell the corpus luteum to continue to survive, so that when it continues to survive, it will continue to produce estrogen and progesterone, and both these two hormones will prevent the endometrial wall from shading. However, if the human chorionic gonadotropin does not go and prevent the corpus luteum from becoming the corpus albicans, what happens is that the endometrial wall will shade, and thereby that person is going to lose her pregnancy because of spontaneous abortion. So, like we said, that the zygote that has become hollow, because the cells in the inner core cells will move to one side, which is the pole, and that pole will indicate the point of implantation or the direction of implantation. And when they start the implant, what happened is that the trophoblast, which are the external layer of cells, will break down the endometrial wall. As they are breaking down the endometrial wall, human chronic gonadotrophin is being produced, and it goes to act on the corpus luteum. So, and then implantation continues to occur, such that implantation completes happening at the end of day 10, with an indication of what is referred to as the implantation plug. So this implantation plug will indicate that that is the site of implantation. And then what happens is that the trophoblast, when it interacts with the endometrial wall, will become two-layer structure. One is the same cytotrophoblast, and the other one is the cytotrophoblast. These two layers are very important because they give rise to the future part of the placenta, while the endometrial wall that has been broken down and while the other ones that are being taken up, the nourishment from the broken cells become what is referred to as decidual cells. These decidual cells will form the maternal part of the placenta. So as that is happening, the cells in the inner core or the inner cell mass will differentiate to become bilaminar discs, that is two-layer discs. So you can see it here. You can see one layer and the next layer on the bottom. So this bilaminar disc, the upper part is called the epiblast. This epiblast is very related to the amniotic sac, while the hypoblast is related to the lower bigger sac, which is the yolk sac. So this is very important because it provides the nourishment for the growing embryo at that time. So later on, at the end of the third week, at the beginning of the third week, these two-layer structure will start stimulating to give rise to what is referred to as the three-layer structure, where we have the ectoderm. So the most upper layer, which is the epiblast, starts to send some of its cells, the cells will detach and then migrate through a narrow pit-looking structure formed. That pit-looking structure formed is the primitive groove and primitive pit. So when the cells that detach from the epiblast migrate into that particular area, they come and lie between the epiblast and the hypoblast, and gradually they come to fill that space. So as they fill that space, they now form like a sandwich, like a bread, butter, and bread at the bottom, where the bread on the top is now our ectoderm, the bread in the and then the butter in the middle is going to be our mesoderm, while the bread at the bottom is going to give rise to our endoderm. So at this point, we have what is referred to as a trilaminar disc. So this is what we have, and you can see the blue layer on top here is the ectoderm. In the middle, like a sandwich between them, is the mesoderm, and the yellow layer there is the endoderm. So above the ectoderm we have the amniotic sac, and below the endoderm we have the yolk sac. So just like we explained, we can see this is a summary of what we just saw, where the sperm cells fertilize the egg, and then in the ampulla, so here you can see ovulation, where the tiny egg is being released by the ovary and is picked up by the fimbria, which is the part of the fallopian tube, while the sperm cells tends to fertilize the egg at the ampulla. And at the ampulla, we are going to start having the division, mitotic division of the zygote. So you can see 2 daughter zygotes, 4 daughter zygote, and 16 daughter zygotes. So at this point, the zygote is now referred to as a morula, and then gradually it becomes a blastocyst. A blastocyst, this is because all the cells in the inner core cells move to one pole, creating one space filled with fluid, and externally have one layer of cells that is going to initiate implantation. So by day 7, we have already started implantation, which could also lead to the release of the human chronic gonadotrophin. So these are some of the hormones that are needed to maintain these particular implantations. So we have the progesterone and estrogen, which are released by the corpus luteum. So the corpus luteum is found inside the ovary, and we need it to continue producing, so continue producing progesterone and estrogen. And that is why the moment implantation starts to occur, they produce what is called the human chorionic gonadotropin. This human chorionic gonadotropin goes to the corpus luteum and tells the corpus luteum to continue existing, such that the corpus luteum will continue to produce estrogen and progesterone, which will maintain the endometrial wall. This prevents the endometrial wall from shading. Alright, so next we also, another hormone that is important is the relaxin of the uterus, and also we are going to, relaxin is important, which is produced both by the ovaries and the placenta. And what it does, it relaxes the ligaments, especially the pubic symphysis, during birth. So these are other hormones being produced during the late pregnancy, preparing for contractions. Another, the most important, is the oxytocin and the prostaglandins, which are very important because they tend to initiate labor, and also they stimulate the uterus as well as the production of the milk. So as that is happening, our main focus is how this brain is being formed. So the brain continues to form also as that particular ectoderm, endoderm, and mesoderm formed. So in this aspect, we get to see the formation of a notochord. So the notochord was also formed during the migration of the cells to form the mesoderm. However, the mesoderm in the midline are more thicker, they are more rod-looking structure, and they give rise to the formation of a notochord. So this notochord tends to stimulate an ectoderm line on top of it, above it, and the ectoderm line above it is going to hyperproliferate such that they form a thick plate at the midline. So this is a thick plate at the midline, the image below it. And then you can see, and that it forms what is called a neural plate. So this neural plate will continue to deepen, and then that it forms what is referred to as a neural groove, that is stage 2. So the neural groove is a critically bent neural plate, and gradually what happens at image 3 is that the neural tube and neural groove will fuse together on the most posterior aspect, giving off to that lemon green color area, which is going to give rise to the neural crest cells. Neural crest cells are very important structures that they stimulate for the differentiation of various tissues, but for your class we are not going to discuss more than that. So in this image 3, you can see that the notochord has detached itself from the ectoderm. Now it is more the neural tube, it is more a structure of its own, deep as compared to image 2. So now this deep structure is referred to as a neural tube, and this neural tube is important because it's going to give rise to some expansions, and this expansion, so these are some image that helps you to understand that. So as that is forming, what happened is that we're going to have the embryo is also forming and folding. It continues to fold such that the plate looking embryo is now going to be a tube ball structure where the amniotic sac completely covers it. So the topper part of it, the topper cap looking structure, is the amniotic sac, which gradually tends to cover all around the developing embryo after the lateral foldings. And then we are going to have an complete embryo, and then you can see deep to that you have a blue round structure and a tube looking structure, that is the neural tube. So this neural tube is important because as the embryo continues to develop, it also continues to develop where some parts of it tends to expand, while some parts tends to persist as a narrow structure, giving rise to the spinal cord, where the parts that will expand is going to give rise to the brain proper. So Okay, we are going to have three expansions. We are going to have the cranial prosencephalic expansion, then the middle is going to be mesocephalon, and then the lower part is going to be rhombocephalon. The distal part of it will continue as a narrow structure which will give rise to our spinal cord. So the most cranial one is the prosencephalon, and from this image you can see the prosencephalon. So what happened is that you can see the prosencephalon is expanded on the lateral aspect. This lateral expansion and all is going to give rise to what is referred to as a telencephalon. So in this image you can see this pink area, which is the telencephalon, which gives rise to the cerebral hemisphere, and then the middle part. So you can see this image. So this the image at the bottom is showing you pink rabbit looking structure or rat looking structure or bunny. So that is going to give rise to the cerebral hemispheres. The lemon green area is going to give rise to the diencephalon. So this diencephalon are deep nuclei that are found in the brain, and we're going to discuss them in full details later on. And then deep next we have the mesencephalon. So the mesencephalon is important because it's going to give rise to the midbrain, while the rhombencephalon, the cranial part of the rhombencephalon, will give rise to the metencephalon, which is going to be the pons and the major part of the cerebellum, while the lower part of the rhombencephalon will give rise to the melencephalon, which will give rise to the medulla oblongata. So after we have this particular structures forming and the dilatation of the brain tube, we're also going to have the flexures, okay? Because of the growth of the cranial part of the neural tube is going to fold so that the cranium will accommodate the large development of the brain. So we are going to see the cephalic flexure. This is occurring during the cranial folding, and then later on we are going to see the pontan flexure, which is important because it needs to separate the melencephalon and the metencephalon from the myelencephalon. It's important. And lastly, you're going to have the cervical flexure, which separates the medulla oblongata with the spinal cord. So this is also an internal view and an image explaining this, but this is mostly in a rabbit brain, and this is also, we can see the different stages of development. So these are the fates of the different parts of the brain that is developing. So you can see the telencephalon, which is a part of prosencephalon, will give rise to the olfactory bulb, hypothalamus, and cerebellum. And then we can see the diencephalon, which is also part of rhombencephalon, will give rise to the retina, epithalamus, thalamus, and hypothalamus. Then we have the midbrain, and then we're going to have the metencephalon, which is going to give rise to the cerebellum, which is the small brain, and also the pons. Then the myelencephalon, which is the lower part of the rhombencephalon, will give rise to the medulla oblongata. Then when we talk about the development of the spinal cord, more of the development of the spinal cord is the arrangement of the neurons in the spinal cord, where we have in the image you can see A and B. So gradually the spinal cord developed from just one single layer structure to, in fact, not just a single layer structure, but rather a 3-zone area, where more immediately after you have the ventricular zone, which is very close to the central canal, or the germinal zone, and then we are going to have cells growing in this particular ventral zone, which is a single layer. They detach themselves and go into another zone referred to as the mental area. So majority of the cells in the mental area are going to form the gray coloration of the spinal cord, while the myelination of covering by the Schwann cells of the axons of these neurons is going to give rise to the marginal layer, which is being explained in the different slides. So you have the inner ventricular layer, then you have the second layer, which is indicated in B, which is called the mental area, and also you're going to have the marginal area, which is also indicated in B, which is the last area. Then also the expansions, because of we know that the spinal cord has expansion where we have the posterior aspect and the ventral aspect. So we'll have the anterior roots. That particular plate is going to expand such that you have what is referred to as the alar plates, and also the posteriorly this is referred to as the alar plate, while ventrally is called the basal plate. So as the brain is developing, it is important to know the critical time of the development of the brain, which is indicated by this image. Is showing that from conception, which is day 0 to day 4 to week 4, neurulation is occurring. So neurulation is basically what we just explained, how the formation of the ectoderm, neural plate, neural groove, the folding of the neural tube, dilatations of the neural tube all occur within this particular 4 weeks. And then you also have the formation of the neurons, which is called neural proliferations, continue to extend all the way. And then you also have the important neural migrations, where neural cells tend to migrate from one point to the other to populate that particular area. So especially in this particular first trimester, that is one, what happens is that there's going to be, whenever a reagent, be it physical, chemical, or whatever exposure a pregnant woman is, she might tend to have an increase in the condition or associated with the development of the brain. So any agent or pathogens or whatever it is that is being exposed, or the mother is not taking, like the folic acid, which she's supposed to take during that week of pregnancy, and she's not taking, there is a high tendency of a formation of congenital defect. So anything that could lead to the formation of any abnormal development or abnormal morphology, that substance is called a teratogen, and that morphological malformation is referred to as congenital defect. So this is very important. So some of the clinical conditions associated with abnormality here is going to be the neural tube defects, which we have different classification of this kind of neural tube defects, where we have some defects where you have the extension of the spinal cord into the seal, the skin, where the posterior vertebra is not formed. And then there are also very different cases where the neural tube does not close, and some cases is we have the hydrocephalus, which is a condition where the ventricles are blocked, and so the cerebrospinal fluid is collected in one part of the brain, and because there was no drainage system, it tends to expand the brain, and that is hydrocephalus. Then we have the cerebral palsy, which is a condition associated with the brain as well. So factors effect of leading to neural tube defect closure could be in genetics, environmental factors such as radiation, also maternal nutrition, and also you remember we talked about the importance of folic acid. So folic acid, if it is high, it could be bad. If it is very low, it could also be bad. So to prevent neural tube defect conditions, folic acid supplements is important to be taken, and genetic counseling is also good for that particular baby. So with this we come to the end of today's class. In the next class we're going to talk about the cerebral hemisphere and the brain stem. Thank you very much. Alright, so to the next class, which is the cerebral hemisphere and the brainstem. So the cerebral hemisphere or cortex is this external rough texture structure that makes up the major part of our brain. It's made of gray matter and white matter. So the gray matter are where we find the neurons of the cerebral hemispheres, and and they are more located on the exterior part of the cerebral cortex, and they form the collections of these neurons forms area, which we're going to talk about later on, and they're involved in a particular function. So the deeper structures or the deeper layer of the cerebral cortex is made of white matter, and white matter is by the myelination of the axons. So collectively the axons are myelinated or covered by what is referred to as the oligodendrocyte. So the oligodendrocyte forms the blanket structure, and it is more fibrous in nature. That is why we have the white coloration. So also deep to this particular cortex rough texture area, we can see a deep egg looking structure there that is referred to as a diencephalon. So when we magnify that particular deeper structures, you see that the diencephalon is a collection of the thalamic family. More of them is the thalamus, and the ventral aspect is the hypothalamus. And then we also have the lateral bumpy structure of this egg superior aspect. You can see two rough structures. They are called metathalamus, and then at the posterior part, which is not very obvious here, we have the epithalamus, and then deep down the green particular area, which is called the midbrain, we have what is referred to as the subthalamus. So the brain stem extends from the midbrain, which is this lower green area, all the way to medulla oblongata, and this pink looking structure with several nerves coming off from it. That is the brain stem. So the brain stem is also made of three parts. We have the midbrain, which is the most cranial part of the brainstem. We have the pons, which is this bluish coloration, and then the lower part is called the medulla oblongata. So the medulla oblongata is the last part of the brain, and below the foramen magnum, it extends or continues downwards as the spinal cord. And they perform, all these particular structures perform different function. So just like I've said earlier on, the cerebral cortex is have different functions based on the area. Where we saw the frontal lobe is more of a motor control than the posterior lobe, or the that is called the parietal lobe. And then it's the temporal lobe is involved with language, and the lower part of the temporal lobe is also involved in memory formation. Deep close to the diencephalon, we have structures that are involved with our emotions and regulation of our homeostasis. So if you notice, the cerebral hemisphere is not a smooth structure. It's thrown into folds and elevations. These folds and elevation allows the brain to stay completely inside the cranium, because if straightened, the brain is a very large structure, so they need to go into the folds to accommodate all the parts of the brain containing the vital information. So the folds, the elevation here, it's what we refer to as the gyri. That is a plural gyri. One of it is a gyrus, that is Gyrus. Then here is a sulci. The depressions are called sulci. Then we're going to see major depressions later on, which are called the fissures. But these smaller depressions are called sulci. That's the plural. Sulcus, that's the singular. And here you can see one on the lateral aspect. It's called the fissure. So we have three major fissures and the brain when we talk about it, and that is the cerebral hemispheres. One, we have it between the temporal lobe and the parietal lobe, which is referred to as the sylvian or lateral fissures. And then the one that separates the brain, the right and left hemispheres of the brain, it's called the sagittal suture. Just as seen in this image, this particular depression that separates the blue hemisphere from the orange hemisphere is called the central sulcus. And it is known that, based on study, that the right part of the brain, which has a function of artistic awareness, creativity, and all other fun things, and it is controlled by the right hemispheres, and people that are left-handed are mostly having the contribution coming from the right hand side, while people that are right-handed have mainly the left hand functions, which involve analytical thought, logistic, language, reasoning, science, etc. So when going back to the different parts of the cerebral hemisphere, we have mainly four lobes, but then we have a new lobe which is found deep to the temporal lobe, which we're going to see. It's called the insular lobe. It's located deep to the temporal lobe, and the insular lobe it's located very deep, and it has several functions. So for these main lobes, we have the dark blue coloration found in the ventral aspect as the frontal lobe, and the lemon green area is called the parietal lobe, and then we have the occipital lobe and the posterior aspect, which is a help to interpret vision. The temporal lobe is this lilac color or light purple color. It is involved with our auditory and senses, hearing, and on the lower part is involved with our memory, that it houses the hypothalamus. The hypothalamus is involved with memory. So the like we said, the insula, we have the sulcus and gyri, and we have major ones that you are supposed to remember in your class. We have hundreds of them, but for your class we are going to just focus on few of them. So one of the most common sulci is the central sulcus, like seen in this top image, which separates the anterior lobe from the posterior lobe. And then you can see another gyri, which is very deeper, has a deeper pink coloration. That is referred to as pre-central gyri because it is in front of the central sulcus. Then the one just immediately behind that pre-central sulcus is referred to as the post-central gyri. So the pre-central gyri is involved in motor innervation, all right, somatic movement, that is movement that we control, while the post-central gyri is involved with interpreting somatosensory. So our touch, pain, and temperature is relied on this particular post-central sulcus. So ventral to the pre-central sulcus, the lighter pink area is called the frontal lobe proper, and posterior to the dark blue color is the parietal lobe proper. And then on the posterior aspect, we have this green area, which is our occipital lobe, like we said, is important for our side vision. And then on the lateral aspect, it is our temporal lobe. So the image below also depicts the same thing. And then on the lower aspect, you can see we have the pons. Here the midbrain is not visible because it's located deep to the temporal lobe, but on this lateral view we can appreciate the pons on the bottom aspect, the pregnant looking area, and then the lower aspect is also the medulla oblongata. These are all part of the brainstem. They're situated behind the brainstem. We have the cerebellum, which is a small brain, and the upper image is shown as an orange structure. All right. So next is we tends to magnify what the sulcus and gyri mean. So sulcus is the depression, while the gyri is the elevations. And then here, if you notice the gyri, the most external or most superior layer is grayish in nature, while the deeper layer is whitish in nature. So the grayish in nature is where we have the cell bodies of the neuron, while the whitish area is where we have the axons of the neuron that are being covered or blanket with the myelin, which is coming from the oligodendrocytes. All right, so here this is another name for the central sulcus, the Rolandic fissure, which separates the frontal and the parietal lobe. And then also we get to see the lateral sulcus or the Sylvian fissure, which is not seen on this image, but that is the one that is located on the lateral aspect of the between the temporal and the parietal lobe. Here we get to see the one that separates the right and left hemisphere is referred to as the longitudinal fissure or sagittal fissure. Then we have another another major one coming back here, separating the parietal lobe, this particular big most posterior aspect, and the occipital lobe, that is called the parietal occipital sulcus, which separates these two lobes. And then the major gyri also you're supposed to focus on is one, like we said earlier on, the precentral sulcus. Gyri is located anterior to the central sulcus, and the major function, like we said earlier on, was the somatomotion. So motor motions that we controls muscles that we can actually voluntarily move. Then we have the postcentral sulcus, which is it is located behind the central sulcus, responsible for a body sensory information. Then we have the superior, middle, and inferior frontal gyri, which is found in the frontal lobe, and these gyri are located in the frontal lobe and are involved in various cognitive functions and also decision making, planning, and execution. So we have the superior, middle, and inferior temporal gyri, which is found on the lateral aspect, and these are mostly involved. So the most superior temporal gyri is involved with auditory processing, language comprehension, and the inferior temporal gyri is involved with memory. That is where we find the location of the hippocampus and the hippocampus gyri. Then we also have the precuneus and the cuneus. These are gyri located within the parietal lobe, and they are involved in our spatial awareness, self-consciousness, and also memory. So here, this is also another way of writing these functions of the frontal lobe. We've seen the motor cortex, that is the one we saw on the most frontal aspect, and we also have the premotor cortex, which is the frontal area that is involved with our planning and coordination, just the thought of it. And then we have the prefrontal cortex as well. So these are responsible for making higher decision and solving problems. It's also involved. It is for our personality. Then we have a Broca's area. This Broca's area is involved in speech production, which is found at the junction of the superior area of the temporal lobe, where you have speech in this illustration, or in the image you can see speech there. That area is referred to as the Broca's area, and then you can see at the junction of the temporal lobe and the parietal lobe, you can see hearing, and you can see memory processing and emotions. So emotions is the one on the most bottom aspect, which is where you are going to find the insula. So the insula, like I said earlier on, is a neural lobe, and then it is very small and located deep in between the parietal lobe and the temporal lobe, as seen in this image where we forcefully open to see the deeper structures. So the functions of this insula is also important. It has a wide range of function because of the location. So before we start talking about its function, let's see the functions of the parietal lobe, that is the lobe at the top of the brain here. And so the parietal lobe, the most common function is that, you remember, we have the postcentral gyri, and so it is involved with somatosensory, which is where you process information coming from the skin, muscles, and joints. So ones coming from these joints are referred to as a proprioception. And also we said that the other parts of the parietal lobe, that is the cuneus and the precuneus area, are involved with our spatial awareness. So here our temporal also is involved with process of auditory and auditory association. You have what is called the Warwick's area. The Warwick's area is the area of language comprehension, while the area of speech is what we call the Broca's area. And also here, also in the temporal lobe, we have what is referred to as the hippocampus, located in the lower part of the temporal lobe, which is involved with our memory. All right, and that memory formation, so both short-term and long-term memory, are formed by the hippocampus. So here is the functions of the occipital lobe, all right, mostly for visual and also recognition of structure areas and objects, as well our balance associated with the vision. Lastly, we have the insula, which is this particular green area, and it is located deep and plays a crucial role in several cognitive functions and emotions. Just like we said, the location is because of the location. So some of these key functions of the insula include sensory integration, where it involves the perception of our taste. So gustatory perception is a perception of our taste. It also has somatosensory perception involving pain, temperature, and touches as well, and also emotion processing. Because of the location of the insula, deep to the insula, we have a structure of the brain known as the amygdala or amygdaloid body. So the amygdala is a structure that helps in processing our emotion, where we get to feel the empathy, social cognition, and self-awareness. So it also gives us our personality. Other functions of the insula based on its location and because of its proximity to part of the diencephalon, which is the hypothalamus, it also helps in homeostatic regulations. Homeostatic regulation involving our autonomic nervous system, which controls our heart rate, blood pressure, and also respiration. That is our vegetative centers. And then also the insula function in our decision making and reward centers. So sometimes when you do good something and the body say very good, automatically without control you start to clap for yourself, because we know that clapping for ourselves is a reward activity. And also it helps us with our consciousness and with our emotions as well. So clinical application to this cerebral cortex involve is neurodegeneration, where where we have stress induced, and then we have lack of enough blood coming to that place. Over activity of the brain could also lead to what degeneration of cells, or sometimes excessive activity or other associating neurodegenerative diseases could stimulate or reduce the performance and also the morphology of the neurons of the cerebral hemispheres. So all these are through different stages where you have oxidative stress, mitochondrial damage. And when you have a mitochondrial damage, it means that there's not going to be enough energy for that cell to continue its action potential. We have neuroinflammation and also cell death, which is also referred to as apoptosis, and these could lead to several disorders such as the Alzheimer's. So the Alzheimer's is a condition where people tend to forget, and they do not have, or there are different types of range of Alzheimer's. And then you also have to have problem with retrograded memories as well as short-term memories. And when it's more advanced, and it is associated with other kind of medical condition, that is when it becomes like a dementia condition. So Parkinson's is a disease where we have tremor, we do not have self-control of our underlying muscle control or muscle tone, and it is associated with injury to the brain stem. And also we might have stroke. We have different type of stroke, and we can see that we have hemorrhagic stroke and ischemic stroke. So hemorrhagic stroke is where there is a blood leak into the cranium, and also those can induce some damages to the different parts of the brain. One of the most common hemorrhagic stroke leading to hemorrhagic stroke is either trauma or aneurysm. So aneurysm of recent is getting very common, and researchers need to study it, especially in Nigeria. Well, ischemic stroke is a lack of blood supply due to the blockage of the blood vessels, either by a cholesterol plaque or blood clot found inside the blood vessels. So those are what leads to some of the common conditions. Other conditions could be mostly the trauma and the shaking of the head, and here are some of the summaries. So here we're going to start talking about briefly about the brainstem. So the brainstem is pons, the midbrain, the pons, and the medulla, just like seen in this image, where you have the green area downwards. So the upper part of the green area that is not looking pregnant, that has a bumpy structure in the posterior aspect, that is the midbrain. And midbrain is very important because it helps us in controlling our smooth motor movement, while the pons are important because they tend to serve as an area where we get to change hands, like a battlefield baton, a relay race. And then we have the medulla oblongata, also containing some important collections of neurons and called the nuclei. So here, like we've seen in this image earlier on, you can see the midbrain. Also, midbrain very important because it has some vital information, especially on the posterior aspect, where you see these two bumpy structures looking. These two bumpy structures in the posterior aspect, the bluish greenish color, is what we refer to as the superior and inferior colliculus, and they are involved in a relay where pattern and exchange for the visual and auditory information. So the visual one is coming from the cerebellum, which is the small brain, while for the inferior colliculus is coming from the auditory system, which is the inner and the ear meatus. And then on the lower aspect, we get to see the pons, the medulla oblongata, which also have several nerves leaving it as the first image. While the second image is a posterior view of also the brainstem. Here, not the dark purple, but the lighter purple just above the green is the posterior surface of the midbrain. So the egg-looking structure on the superior aspect of that midbrain, the posterior midbrain, is what we refer to as the thalamus. The thalamus is an egg-looking structure found on top of the brainstem, and it is involved in our sensory relay station, where sensory information go and relay, and then later on they get projected to different parts of the brain to take in the sensory information from different parts of the brain. All sensory information come into the thalamus except the sense of smell. Sense of smell does not come to the thalamus, but all other senses come into the thalamus before they get projected to different parts of the cerebral hemispheres and also parts of the brain. Next, on the lower arm, so in between the two thalamus, we have this projection that goes downwards a bit, if you notice it. This is called the pineal gland, and the pineal gland is very important because it helps to regulate our sleeping cycle, which you call the circadian cycle, producing the melatonin that allows us to sleep when it is dark. So on the lower aspect of that is where we see that particular midbrain. We can see it has four bumpy structures on the posterior aspect. These four structures are called the corpora quadragemina. There are four of them. That is the quadratus in this particular name. So corpora quadragemina, and then they are either called superior colliculus and inferior colliculi. So these are the major prominent structures on the posterior aspect of the midbrain. Then when we come to talk about the green area, this is the posterior aspect of the pons, where we had the location or where the cerebellum, which is the small brain, sitting. However, in this image, the cerebellum has been removed, and then you can see these bumpy looking structures here. So these bumpy looking structures on the posterior aspect, on the lateral side or the side are called the peduncles. So peduncles are basically arm or pathways. So they allow for the communication coming from the pons to the small brain. So these depression found also on the posterior aspect of the pons is where we find the location of the fourth ventricle, but because the small brain, the cerebellum, has been removed, that is why we get to see the floor of the fourth ventricle. Then here on the lower aspect, the pink area, it's called the medulla oblongata, and we can see it is a rough particular structure, and this is because it allows for the movement of a bundle of myelinated fibers coming from the brain, spinal cord to enter into the brain. Alright, so this is also an image showing that, and this is a real life where the middle a middle ped is also referred to as the middle peduncle. So it's showing that is the posterior aspect. That is, we're talking about the image A here. The black background is also similar to the image we see on the left hand side. So this one is a real life midbrain stem, and then on top there you can also see the thalamus, and then below it you can see cerebral peduncle. This one is a small brain peduncle right here. Then you see the red nucleus. The red nucleus is a very prominent structure found in the midbrain, which you're going to see in the next slide. And then you also get to see some of this important structure. On the posterior aspect, you can see what is referred to as inferior colliculi or colliculus. And then as well on the posterior aspect, you will get to see lateral gene body. So lateral gene body means lateral geniculate body, and then you have medial geniculate body. These are all parts of the thalamus, which talked about earlier on. The pumpy structure found on the thalamus. All right, so this is just to show you that the real life structure is also very, very similar to the illustration we are showing. So when we talk about the brainstem, this is a cross section through the brainstem. So some of the important structures into and the brain stems are, we're going to talk about just the major important structures. So the image on the left hand side, where you can see, and these the structure we saw earlier on, is showing you the ventral aspect of the brainstem and the posterior aspect view of the brainstem. And when magnified, the images below it, you can see what the structures are found in the brainstem area. So in the brainstem area, you can see these blue small nerves coming off from the ventral aspect below a breast looking structure. That breast looking structure is referred to as the mammillary bodies, and then you can see it is very tiny and narrow at this particular structure. So we're still discussing the image on the left hand side. So that is the narrow area, or the narrow neck is the whole part of the midbrain. So superior to the midbrain, you can see we have the optic chiasma, and inferior to it we see the pons. So on the ventral aspect, you see one blue pair of blue structures leaving the ventral aspect. That is the oculomotor nerve, the nerve that controls the muscles of the eye. So all we have 7 muscles in the eye. All the muscles except 2 are controlled by oculomotor nerve. The other one is one of the muscle in the eye is controlled by the oculomotor nerve. So the trochlear nerve. So the trochlear nerve is a nerve coming off from behind. So if you notice, just below that particular breast looking area on the lateral side, you see another blue structure coming from the behind. That is the trochlear nerve. So the trochlear nerve only innervates the trochlear superior oblique muscle of the eye. The superior oblique muscle of the eye is important because it also does it depresses the eye, it adducts the eye, and also medially rotates the eye. That is what it does. So this is the posterior view. The lowest image here is a posterior view of the midbrain, and you can see that we have the superior colliculi. In between the 2 superior colliculi, we have the pineal gland with the projection, a tiny projection of structure, that is the pineal gland. And below the superior colliculus, you have the inferior colliculi, and then below the inferior colliculi, you can see the blue structures winding around. So this is the point of origin for the trochlear nerve, and then gradually winds around and comes ventrally and goes to innervate that superior oblique muscle. So the image on the right hand side is a cross section of the brain, the midbrain, where we appreciate some of these deeper structures from the dorsal aspect of it. So here is just indicating the cross section of the midbrain. So this is a cross section of the midbrain. So the dorsal part of this midbrain is referred to as the tectum. So this tectum is referred to as the tectum while the ventral part, okay, the remaining ventral part is called the tegmentum. It is not shown in this image. So the tegmentum. So the tegmentum is the anterior part of this image, except where it stops at this gray looking structure. This gray looking structure or dark brown looking structure is what is referred to as the substantia nigra. The substantia nigra, just like the name implies, is a dark area in the midbrain, and it helps in controlling that muscle movement, the motor muscle movement. So if there is a lesion in this or degeneration of these neurons, these dark looking neurons or the substantia nigra cells, one is going to end up with a condition known as Parkinson's, among other kind of conditions, but the most common condition is the Parkinson's. Another condition associated with this degeneration is genetics. It's called the Huntington condition. So here in the tectum, mostly what you find is where you see the superior and inferior colliculi. Then just ventral to it, you get to see the cerebral aqueduct. This central aqueduct connects the third ventricle with the fourth ventricle, so it's very important. It needs to be open all the time. Blockage of this particular aqueduct or duct would result to hydrocephalus, and we do not want that. We want the cerebrospinal fluid produced in the ventricles to move around and exit, and also we want to constantly be supplying and also draining the cerebrospinal fluid. It's important to maintain the structure, maintain the cycle of collection of waste and supplying of nutrients to the brain. So just surrounding this central aqueduct, we have what is called the para-aqueduct gray matter. It's very important because it helps us in maintaining perception of pain. So if you have a lesion in that particular area, you're not going to feel pain. So this is important for that perception of pain. Then next we have the reticular formation. So the reticular formation is a kind of formation or collection of nuclei as well as white matters that is involved with our consciousness, we are aware, and hemostasis. That is the area that helps in communicating with our special centers, that is respiratory centers and cardiovascular centers. It helps us being alert and awake. So most ventral, you have the red nucleus. The red nucleus is very important because it also helps us in controlling our motor movement. Then we have more ventrally, we have the substantia nigra, and the substantia nigra have two layers. We have the pars compacta and pars reticulata. All right, so later on we're going to go deeper into pars compacta and pars reticulata layers of substantia nigra when we talk about the basal ganglia. So the most ventral parts of these of the midbrain is what is referred to as the crus cerebelli. So this crus cerebelli is important. So this is mostly seen in this image where you see the crus cerebral peduncle. So the cerebral peduncle or the crus cerebelli, it contains the matter, white matter or myelinated axons coming from the motor area, and their intention is to go down to the spinal cord to innervate or control the movement, our motor movement. So motor movement is voluntary movement, while some of the fibers also coming down here will exit and go to control the muscles of the face, so that such as our smile and other muscles controlling the eye. We can actually turn our eyeballs based on this control. So here also you can see ventrally one nerve is living at the midline. That nerve is called the oculomotor nerve. That is the nerve I said that innervates most of the muscles of the eye, except the superior oblique muscle and also the lateral muscle of the eye, which is called the abducens muscle. So only these two muscles are innervated by different nerves. All other muscles in the eye are innervated by oculomotor nerve. So here are just the functions of all the structures. Most of the important structures we see in the midbrain. So we have the tectum, which is have the superior colliculus and inferior colliculi, and also we also get to see the tegmentum. So the tegmentum is a ventral part where we get to have the substantia nigra, which we talked about. Is important because it produces dopamine that controls our motor movement and also reward centers, especially when you dance. You tend to be happy with the execution of your dance, etc. The red nucleus is also important for the coordination of motor, muscles, and muscle tone. All right, and then we have the reticular system, which also regulates our arousal, sleep awake cycles, and our tensions, which also, you remember when we talked about our hemostatic centers, where we have the cardiovascular system and also respiratory system. The periaqueduct gray matter, like we said, is involved in modulation of pain and defensive behaviors. So now the cerebral peduncle is the most ventral part of these midbrain, and like I said, it contains the tract going to the spinal cord and other parts of the brainstem, as well as going to the to innervate the face. So the functions of the brainstem, just like we said, it does motor control, sense process, pain modulation, arousal, and consciousness. So if we have a medical condition associated with the brainstem, like I said, one of the most common syndrome is the Parkinson disease syndrome, and also is going to affect the movement of the eye because of the superior colliculus, and also impairment of hearing because of the inferior colliculus. All right, so next we have what is referred to as the pons. This is a cross section of the pons. So where you see the rhomboid fossa here on top of the image A, rhomboid fossa, that is the posterior area. That is where we find the fossa of the ventricle, of the fourth ventricle. So you know that we're going to see the third ventricle found in the brain around the diencephalon, and then it communicates via a narrow opening via the aqueduct of Sylvius, and then it comes into the fourth brain, which is and the fourth ventricle, which is located in the posterior aspects of the pons and the ventral aspects of the cerebellum. Just like seen on C, this is the posterior view, where you have the median sulcus, facial colliculus, and also striata medullaris. These are all found at the floor of the fourth ventricle. So here B is just a ventral view of the pons and also the medulla at the bottom. Medulla aspect is the one that is more smaller than the superior aspect. So where the two parts meet, the pons and the medulla meet, it is referred to as a pontomedullary junction. It is very important because it contains several nerves coming up from that particular area. So this image also, you can see the first image on the left hand side is trying to magnify the level of the pons, where it is showing the ventral and posterior view. When magnified, you can see the pons, the ventral aspect, which is the first image on the left hand side. The image of the pons, which is the ventral view of the pons. Superior to the pons is the midbrain, which has been removed in this image. It is not very clear, just lower part of this is visible. So the pons, you can see it has and the lower communication with the medulla, and you can see several nerves coming out from the point of junction between the pons and the medulla. So from the midline, we have what is called the sixth cranial nerve. The sixth cranial nerve is the abducens nerve, which goes to innervate that other muscle that controls the eye that was not innervated by the trochlear. You remember we said the abducens. So this particular nerve goes to control, it's the abducens nerve, which goes to innervate or control the lateral rectus muscle of the eye, which helps us to look sideways like that, side eyes. Okay, so it is the abducens nerve. Next, on the lateral aspect to the abducens nerve, the green nerve, it is the facial nerve. The facial nerve is involved in the control of the muscles of our facial expressions, so that is why it's called the facial nerve. This next nerve, more lateral, is the eighth cranial nerve, and it is called the vestibulocochlear nerve, and that is why you see it looks like two bundles of nerves. So one part of the vestibulocochlear nerve. So that is the vestibular part of the eighth cranial nerve, is involved with our balance, and while the cochlear part of the eighth cranial nerve is involved with our auditory, the recording of the impulses coming from the auditory membrane, the bones of the ossicles, and then finally onto the organ of Corti, that is the organ of hearing in the adult human being. So all these three nerves are coming from that particular area where the pons and the medulla fuses together, and that is what is referred to as the pontomedullary junction. So you can see how important the pontomedullary junction is. So another nerve, very big nerve found in the middle of the body of the pons, is the fifth cranial nerve. The fifth cranial nerve is trigeminal nerve, and it needs several points of origin because it controls both several smooth and deeper muscles of the face and neck, and that is why it is called the trigeminal. The reason it is called germinal is because it has going to have three branches. So the first branch is called the ophthalmic branch, which helps on controlling the eye area, and then we have what is referred to as the maxillary branch, which controls structure found at the middle of our face, the upper part of our teeth, the lateral part of our nose. All these are called the mandible and the maxillary area. Then the mandibular area are the last branch is controlling structures found on the mandible layer, that is the lower part of our jaw. All right, so the image on the right hand side is a cross section through the brainstem and showing the inner structure of the pons. So most importantly, we have the corticobulbar and corticospinal tract, which is found in the more red ventral aspect. This is the purple structure in this image. So this contains the fibers that goes to control the motor muscles of our face. So corticobulbar is motor muscles of our face, while corticospinal is the one that goes to control the motor movement of the muscles of our other parts of the body. So another common structure seen here is the middle cerebellar peduncle. This is different from what we saw in the brainstem. The brainstem is the cerebral. This one is the cerebellar. The peduncle is a communication between the small brain and the pons. Then we also have other nuclei in the brainstem, but I don't think you need to remember all these smaller nuclei. So based on that, the pons is divided into two parts. We have the ventral pons, which contains large bundle of nerve fibers connecting the cerebrum to the cerebellum. You remember the connecting the cerebral hemisphere and the small brain, and houses several pontine nuclei. So the dotted blue nuclei, and by the side of this purple nuclei are the pontine nuclei, which relay information from the cerebral cortex to the cerebellum for coordination and control. So the cerebellum is mostly a part of the brain that helps in coordinating and also balancing all the movements, so we do not hyperextend or hyperflex our muscles so that we do not fall. So it regulates the tone of our muscle contraction. So the dorsal part of the brain, the pons, is involved with mostly, we said, is the floor of the fourth ventricle as seen in this image, the lower part, the two image on the lower part, the one on the right hand side. And also it contains the reticular formation, just like it also contained in the brainstem. So the reticular formation all extends from the brainstem down to the pons, from the midbrain down to the pons as well. And like we said, it regulates our sleep, wakefulness, and arousals, and also contains the nerve area. So do you remember we said we have those nerves of facial expression and other functions. All right, so these are some of the important structures in that particular area. So based on that, the function of the pons includes motor control. It also assists the brainstem, as well as the cerebral cortex, the cortex of the big brain to also control the motor movement, so that when we are moving our left side, when we are walking, our left side is balanced than the right hand side, so that we don't fall off. And also it helps in sensory processing, and this sensory processing includes the hearing and also the balance, which we have the vestibulocochlear nerves doing. We also have autonomic functions, you remember, based on the reticular formation, okay, to help in regulating the breathing, the heart centers, and blood pressures. We also have the sleep and arousal, also via the reticular formation, and also facial innervations, just like the nerves that are leaving that particular area. So if there is a lesion in this particular area, it's going to give us rise to the symptoms of stroke, where we have our speech is slur, we have numbness, we have nausea, okay, because of those centers we have inability to we will not be able to have a very good balance. We have double vision, loss of coordination, and a lot of other symptoms. So then lastly, when we talk about the brainstem, we are talking about the medulla oblongata, which also from this image you can see this is a pink area of the brainstem, and when expanded, this is the point we see. So you can see the medulla oblongata is majorly made of several fibers of the nerve coming off from it. However, these are just containing only 3 nerves, and unlike how it is being portrayed here. So the ventral aspect of the medulla oblongata, like we said, contains very much of nerves, as compared to what we see on the posterior aspect. This is the lower image on the right hand side. So the ventral image found on the midline, you're going to see we have a median fissure, which is a depression, a very slight depression, and just lateral to the depression on both sides, you have this a pyramid looking, like a pillar looking structure. That pyramid looking structure is what referred to as the pyramids of the medulla oblongata, and these pyramids of medulla oblongata contains the cortical spinal fibers, the fibers that controls the motor movement, coming from the cerebral cortex, came down to the midbrain as the crus cerebelli, came down through the pons as the cortical pontan, as well as cortical spina, and now the cortical spina is descending down into the spinal cord via these pyramids. And one fascinating thing about this is that most of the fibers, that is about 80 or 85, depending on the textbook you read, most of the fibers from one side of the pyramid crosses to the opposite side. So it means the opposite side of the brain is the one controlling the side of the brain. For instance, the right side of my body is being controlled mostly by the left side of my brain. That is basically what it means. And as well, the left side of my body is controlled mostly by the right side of my brain. So that is why majority of the fibers coming downwards to control our muscle movement via the corticospinal tract are going to cross to the opposite side at the level of this pyramid. So this crossing is called the decussation. It's called the decussation. All right, so this is a pyramidal decussation where fibers of the corticospinal crosses to the opposite side. So immediately lateral to the pyramids, we have a bundle of nerve, which is referred to as our hypoglossal nerve. The hypoglossal nerve is our 12th cranial nerve, and hypoglossal is meaning it's controlling and the structure of the tongue. So anytime you hear the glossus, is also talking about the tongue. Then lateral to it, you also have another bumpy structure. That bumpy structure is referred to as the olivary nucleus. The olivary nucleus is also very important when it comes to our balance and coordination, which communicates or send fibers directly to the cerebellum, that is the small brain, and maybe we'd also get to look at that briefly. So on the lateral aspect, on the upper part, the green fibers coming off are the glossopharyngeal, so glosso also to the tongue and also to the pharynx. That is why the name is glossopharyngeal, and it is our 9th cranial nerve. Then below the 9th cranial nerve, we have another blue nerve coming off from different parts, but they fuse to become one single nerve. That is our accessory nerve. The accessory nerve is also a nerve that goes to innervate one muscle, which is the trapezius muscle found around behind the neck. All right. So the medulla oblongata is divided into three main parts, like we saw earlier, the ventral part, and now we're going to be seeing the dorsal medulla. So the dorsal medulla most of the time contains several nuclei and also a deep reticular formation. Remember the reticular formation that crosses now to the medulla oblongata, the pons, and also to the midbrain, and is involved with our awareness status and consciousness status. Don't forget, we also have those brain centers as well as respiratory centers located also in the brainstem. So apart from that, we also have several nuclei that are important for different communication, as when we see the ascending and descending tracts coming into play. All right, so this is what we get to see. One of these important nuclei is called the solitary nuclei, and the solitary nuclei is a nuclei that we are going to see when we start talking about the sense of taste, that is the gustatory pathway, and you're going to see some of the fibers coming from the tongue coming to this, what is referred to as the solitary nuclei. So it's important. It is very important, and some of this vital information it contains the cardiovascular centers, like we said, regulating the heart rate and blood pressure. It also contains the respiratory centers, also involved with the breathing and rate and as well depth. We also have the vasomotor regulatory centers, which affects the blood flow and blood pressure as well. So also sense some of the sensory and motor function doing here. It processes sensory information coming from the face and neck. So like we said, it processes also information like the gustatory pathway, which is for the sense of taste. So it controls swallowing, coughing, and also our vomiting centers are also found in the medulla oblongata, and they also release motor signals to the muscles and of the face and neck and shoulders, which are the cortical bulbar tract. Okay, so here is a cross section into the brain, where on the ventral aspect you see the pyramids, just on the lateral aspect you see the inferior olivary nucleus, and then deeper so you can also see the medial lemniscus, which are also involved with our centers, and also on the posterior aspect you start to appreciate several nuclei which you do not have to remember in this program. And the posterior aspect you get to see what is choroid plexus, and they are found more located inside the fourth ventricle. All right. So damage to this particular area could lead to several conditions, including the respiratory failure, cardiac arrest, impaired swelling, just like the function, like we listed the function. So we also have the loss of sensation of the face area, and also we have paralysis, facial paralysis, and altered senses. All right, so these are some questions that you can study on your own. And any questions, if any questions, you can drop it in the question box or send it to me. And if no questions, have a blessed day.